this video, we will discuss about the gene regulation and expression. We will focus into the Lac Operon. First, we will explain the concept of operon and gene regulation. Operon is a functioning unit of DNA containing a cluster of genes under the control of a single regulatory signal or promoter. The genes are transcribed together into an mRNA strain and either translated together or separately in the cytoplasm. The Lac Operon found in E. coli induced by the existing of lactose in human colon. It introduced by Jacob and Monod 1961. The background of the study: the E. coli lives in human colon. It breaks down lactose. It's involved in the lactose metabolism, in which it will hydrolyze lactose into glucose and galactose. E. coli uses three enzymes to take up and metabolize lactose, the genes for which are clustered in the Lac Operon. The first gene, LAC-Z, is code for beta-galactosidase, which hydrolyzes lactose to glucose and galactose. The second gene, LAC-Y, code for fermiase, the membrane protein that transports lactose into the cell. The third gene, LAC-A, is code for transacetylase, an enzyme that detoxifies other molecules entering the cell via the permease. Unusually, the gene for the Lac repressor, LAC-I, is adjacent to the Lac Operon. The components of operon: it consists of regulatory gene, LAC-I, and the Lac Operon that consists of promoter, operator, and structural genes LAC-Z, LAC-Y, and LAC-A. The components of the Lac Operon and its function in E. coli. First, the regulatory gene LAC-I contains gene to synthesize the repressor protein. Promoter is the attachment site for RNA polymerase. Operator is the attachment site for repressor protein. The structural genes consist of LAC-Z, gene to synthesize beta-galactosidase, LAC-Y, gene to synthesize beta-galactoside permease, and LAC-A, gene to synthesize beta-galactoside transacetylase. Now we will look into the mechanisms of the operon in the absence and presence of lactose. Without lactose, LAC-I is transcribed and translated into repressor protein. The repressor protein will bind to the operator. This will prevent the RNA polymerase from binding to the promoter. There will be no transcription of structural genes, no enzyme produced, and the operon system is said to be not active or switched off. Now the mechanisms: when the lactose is present, the lactose will be converted into its isomer that will be called allolactose. LAC-I is transcribed and translated into repressor protein. Allolactose binds to the repressor protein. The repressor protein will change its conformational shape. This will cause the repressor protein cannot bind to the operator. The RNA polymerase can bind to the promoter. Then the transcription of structural genes occur. The enzymes are produced. The operon is active or switched on.